Thanks, everyone. Jason Acker, one of the other affiliations I have is with Canadian Blood Services as one of their senior scientists. So a lot of what I'm going to be talking about today reflects a lot of the work that we do at a national perspective to bring blood products from donor to recipient. So much of the technology we're going to be leveraging as we think about where we get these mitochondria from and how do we actually process will be based on the expertise that we've developed in delivering blood products to patients. So why platelets? We've heard it alluded to a number of times today as a source of mitochondria. Why would we consider platelets as a source? We know, for those of you that work in the health space, that platelets are collected by the blood services globally to treat bleeding patients every day in our Canadian hospitals. And approximately 150,000 of them are collected every year by Canadian Blood Services and Quebec to support Canadian patients. On average, an apheresis unit contains roughly 3.5 to 4, I don't know what that is, 4 trillion platelets. And each of those platelets contain anywhere from 5 to 8 mitochondria. So we're looking at a significant pool of mitochondria that are available in a single unit of platelets. And if we're thinking about the question I asked this morning was a dose. How efficient do we have to be at recovering mitochondria from these platelets if all you need is 1 million of them, or 2 million of them, or 3 million as a dose? So right away you can do the calculation and realize we've got a lot of potential therapeutic doses in a single apheresis product. One of the things to consider, and what I'm going to talk about over the next few minutes, is just really, as we're thinking about this source of platelets, what are some of the factors that aren't often understood, except by those of us who work in the transfusion medicine world? One of the advantages of working with platelets is that they're collected and produced in a GMP environment already. They're a regulated therapeutic. They're produced in a GMP environment, a closed system. We store them already in Canada for up to 7 days at room temperature. And we quality control them extensively, and that includes not just quality control of the product, but quality control of the source, and that meaning the donors. So there's an extensive donor screening program that are in place to collect these platelets every day from our donors for our patients. Approximately 10% of the platelets that are collected are discarded annually, so that's roughly 15,000 units annually that are discarded. And the cost of this product is about $650. That's what it costs Canadian taxpayers to collect a unit of platelets and make them available. So 15,000 of those aren't actually being used. They're being discarded. So there's a significant cost to the healthcare system from these discarded products. One of the other things that we also know very well in the transfusion area is that platelets do accrue a storage injury during that storage, and that's why we can only store them for 7 days. That and the increased microbial risk to that product because we're storing it at room temperature. But platelets will undergo a storage age injury as a result of storage, and that's both biochemical, morphological, their responsiveness to agonists and their activation, all important to their ability to function as a viable platelet. We also know, though, that there is decreased respiration, reduced ATP production, increased ROS in these platelets that starts to accrue as early as 3 days of storage. So if we're thinking about when we would use these platelets, if we have to wait till 7 days that we want to use them because they're going to be discarded, those may not be the ultimate time point to use these platelets because of the detrimental effect that's happening to the mitochondria contained within them. That doesn't mean, though, that there aren't ways to mitigate that damage. I really want to put this slide in because not a lot of people realize where platelets come from and how we actually make them available. So there's really 2 ways that we make platelets available in Canada. One of them is from whole blood donors. So this is a blood donor that goes in and donates their whole blood. And those platelets are then isolated through a series of centrifugation and processes that we have at scale in our blood manufacturing sites all across Canada, to produce a pooled unit of platelets. So this is where we will produce typically 5 to 7, pool 5 to 7 Buffy coats to produce a single pooled unit. The other is an apheresis collection process. There's a variety of commercial apheresis devices that we use to collect platelets. Now, the whole blood are obviously a pooled product. An apheresis is a single donor product. And the advantage of the apheresis products, as I'll talk about in just a minute, is the concentration of platelets, but also that we do extensive genotyping of our donors for those apheresis products. So we've got excellent characterization data on each of those donors that are producing those apheresis products, which may become important as we're thinking about them as sources of mitochondria. The other thing that we do routinely in the blood system, and we have variations of these products that we make available, is we will store the platelets cold. And there's an initiative right now in Canada to bring cold platelets to the clinics. We will cryopreserve the platelets. We don't do that yet in Canada, but it's being done extensively around the world, mainly to treat, to support military operations and defense applications. But there is pre-hospital applications for cryopreserved platelets, and PRT platelets, or pathogen-reduced platelets. But not all platelet units are the same. Pooled platelets, which I've talked about in Canada, we've just recently switched to pathogen, or pooled platelets, sorrel and treated, or PPT platelets. This combines 7 Buffy coats with a platelet additive solution called PAS-E that's pathogen treated with a Cirrus technology that uses amotosalen to produce two pooled platelet units. And the quality of those platelets is outlined here in terms of the concentration and volume, so you have an idea of what's in there. And if you contrast that to an apheresis platelet, where we're actually collecting and storing them at room temperature in ACD, or acid citrate dextrose solution, they're much larger volumes and they contain more platelets. So an apheresis product has some superior characteristics if that's what we're targeting. All of them are leukoreduced, meaning we push them through filters in order to remove the leukocytes. Now, Ethan on my team is exploring that filtration process as a way to isolate the platelets, or isolate the mitochondria from the platelets. We do all of this in a closed system. So this is all, we don't need biosafety, we don't need clean rooms to do all of this. This is done in open air in our production facility. So from a manufacturing ease of use and cost, no, there are things that we do in the normal production of a platelet unit that would likely be amenable to what we want to do with the mitochondria. The other factor that I just wanted to highlight again in the few minutes that I do have is that blood donors are unique. And we've been studying this in my team for almost a decade, both as it relates to red cells, but also platelets and other cell therapy products, and how that influences the quality characteristics of those products. And in the case of platelets, we know from extensive work that we've done, but also partners that we work with, that platelets are, the blood donor age and the sex of the donor influences the characteristics of those platelets. And we also know from biology that platelets or mitochondria that come from female blood donors or female individuals are different than male individuals. So how do we reconcile that sex dimorphism as we're thinking about the development of a mitochondrial product? And we have some thoughts about how to think about that. So just quickly in summary, platelets represent a natural, readily available source in high numbers that we can access. Apheresis whole blood derived platelets are discarded on a daily basis, but considerations need to be made for the manufacturing and storage of those products in terms of when we would access them for isolation of mitochondria. But the advantage of having extensive donor screening, closed system manufacturing, all of that is going to streamline the clinical adoption of a mitochondrial product derived from a platelets versus some other method or some other source. But the mitochondrial isolation storage from platelets needs to be considered from this upstream factors. So the pre-analytical, pre-isolation factors, including donor sex and age, will need to factor into our decision as we start to go down this path. So it can't just be simply, no, let's take whole blood from volunteers in our research group, use those platelets, and then try to extrapolate to what's going to be available on a commercial front from a blood supply. We need to adopt what we already know. So with that, I'll look forward to the next speaker.